After fully analyzing your data in the discovery-driven analyses and the differential gene expression analysis and functional enrichment sections, what you can do is move on to the GEO tab. The GEO tab is related to the Gene Expression Omnibus public data repository for sequence data. That process to submit your data to GEO can be somewhat troublesome, and so what we've done is a tool here that can help you in that process. We have a questionnaire that has information related to your data. What we also have done is automatically populated this based on the information that's provided within the server previously. So if you have information related to the number of samples, the sample names, and some of the other information, the limited information that's provided, we try to populate that already, along with some descriptions of how you can actually fill out this form. There's a lot of information that goes into it, but it is set up a little bit better for users to then go through this rather than trying to populate the actual metadata file, which can be quite troublesome. Once you've gone through this entire process, you can download the metadata file, which is an Excel document that is formatted appropriately for GEO submission. You can also download the processed data files, which are separate count files generated from the original count matrix that you had provided previously. Once you've downloaded those two things, the only other piece of information you need then is the actual raw FASTA or FASTQ files, sequence files that you would need then to submit to the Gene Expression Omnibus. If you have any other questions related to this server and how to use it, the More tab has a lot of information, such as the tutorial. It's a pretty detailed tutorial. It also has information related to how you can actually load the server from an R session as opposed to using it in a web server environment connected to the internet. So if you want to run this locally, offline, there's information related here to how you'd actually run that just in an R session. We also have a series of frequently asked questions, ranging from something as simple as what does IRIS stand for, to a lot more complex things like what is PCA and MDS. The About Us section has information related to the people who are involved in this project, so feel free to reach out and contact us if you have any questions. Thanks.